Right, just for a little transparency, we'll talk a little bit about our last exam. Overall, something a little bit closer to a bell curve than what we've had in some of the other exams, 76 average, so I did not curve this exam. We'll just wait and see how the final goes, whether I curve that or not. I just really want to just talk about a couple of questions that were commonly missed. There was a couple of other ones that were less than 50% of you got right, but these were two of the kind of most common ones. This question is really just getting at the steps of phagocytosis. So if I give you an organism that we didn't talk about, chlamydia. It says it can prevent the formation of phagolysosomes. Okay, so that's where the lysosome fuses with the phagosome membrane. So assuming that the organism has already been phagocytized, this is going to keep the organism from actually being killed by those lysosomal enzymes. So keeps it from being digested. And then the second one, what is adaptive, what is humoral, what is cell mediated? Right, so adaptive, humoral means the body's humors. So that's the antibodies that are in the bloodstream. So humoral immunity involves antibodies and especially targets those extracellular pathogens. So things that are outside of the cell. An intracellular pathogen would be inside of the cell and would have to get presented by the MHC1 for the T cells to recognize it. Okay. All right, so again, I haven't had too many people stop by and look over their exam. I have a lot of availability, especially on Thursdays and Fridays. So come by if you have time to do that. Okay, so yes, question. No, you can come back in and look at any, I have them all. So yep, you can peek at any of them that you want. Good, so any questions about the final, any of that information that I gave to you? Okay. I'm thinking that guess of which pathogens we're going to get to, I'm thinking that that's going to be about right. But I'll give you, again, updated learning objectives with certain ones crossed off. There are a few pathogens that were on my original list that we're not going to get into that you will see some homework questions on. So in that way, you at least have to look in the book a little bit. There's only a few of them. So you may see something on, say, tetanus or botulism, for instance, that we didn't talk about, but at least I'm going to make you learn a little bit about it by answering a few homework questions. All right, so we left off last time, we finished up, we talked a little bit about bacillus in general and talked specifically about anthrax. Got kind of rushed there at the end, but we do have a vaccine. It's just not a very good one. Sometimes I think it's technically called an acellular vaccine. It's actually part of one of the toxins. Maybe there's a couple of different parts included in it, but really high incidence of serious side effects with it. So I believe that it's rarely used. Maybe if you worked with anthrax in the lab or something like that, then you would want to have it. We don't even give it to the military anymore, where, that are serving in places where anthrax spores are more prevalent in the soil. Again, it's good but we have a different one in animals, and for whatever reason, the one that we have for humans is not so great. All right, so I want to move on and talk about another genera of gram-positive bacilli, and that's the Clostridium. They're kind of similar to Bacillus. Remember, Bacillus is also a gram-positive rod-shaped organism. It's the other genus that's pretty famous for being a endospore producer. So I think you can't really see this great. This is a picture that I took, I believe, from one of our spore stains in lab. So remember, when you're spore staining, the vegetative cells are red, the spores should take up the green from the malachite green. And when you spore stain Clostridium, when I showed you the anthrax, the spores were centrally located in the middle of the rod, and the Clostridium are terminal. So we get this, end, the endospore is kind of at the end of the cell, and it's usually swelled up a little bit bigger than the cell. So sometimes we call those, tennis racket spores or lollipop spores or something like that. Okay, so that's kind of what they're most famous for. Unlike the bacillus, they are anaerobes. Okay, so where the bacillus are catalase positive, the Clostridium, are catalase negative. So yeah, catalase test, you can use that to tell the difference between staph and the strep, as well as the bacillus and the Clostridium. They are saprobes, just like bacillus, so they're going to feed on dead and decaying organic matter. Interestingly enough, there is at least one species that are considered commensals with humans. So some of us would have C. Clostridium difficile as part of our normal microbiota in our large intestine. Okay, so our colon is one place in our body that's essentially anaerobic. Okay, so these are all either strict anaerobes, all the Clostridium are either strict anaerobes, or kind of, some of them are more aerotolerant. Like there was a species that we used in the lab, that Clostridium sporogenes, I believe. Okay, so there's some really nasty characters in this genus, nasty species, because they kind of are famous for producing very potent neurotoxins. So, for example, we're not going to go into any more depth on Clostridium botulinum, but botulism toxin is one of the most deadly substances known to man. Botulism food poisoning is fairly rare. I think there's somewhere between like 100 or 200 cases of it in the US every year, but it's usually from someone that has improperly canned their home vegetables. So know what you're doing. You got to bring it up to the right temperature and keep it there for the right time in order to kill any botulism spores that might be on those vegetables. Clostridium tetani produces a neurotoxin that keeps our muscles from relaxing. Actually, the botulism neurotoxin works the other way. It keeps our muscles from contracting. So we die of asphyxiation, but the tetanus, it keeps our muscles from relaxing. So we get this rigidity. Both of them are treatable if we catch them early on, but if we have to get a tetanus shot every 10 years or so, or especially if you're traveling to some country where medical care might not be too reliable. Okay, but I thought it seemed appropriate to spend more time on Clostridium difficile, because you're much more likely to encounter that, those of you that are going into some kind of medical career. Okay, and probably some of you have encountered it already. So this is an organism, I don't know what the percentage is of us that have it as part of our normal microbiome. It's not everybody, but a good healthy percentage of us. Of course, this causes a really very nasty diarrhea, sometimes can be kind of a chronic diarrhea. And there are about at least 3 million cases of Cdiff diarrhea. That's just within hospitals every year. And there's probably a lot more than that, where people that don't have to be hospitalized or not already hospitalized. It's considered an opportunist, all right? So if you're healthy, you're not typically going to have a C. diff infection, but actually the number one risk factor here is being on a course of antibiotics. So I have talked to college students that have had C. diff diarrhea and just because they went on antibiotics. And of course, you go on some broad spectrum antibiotic, and that depletes some of the variability of your normal microbiota. And if you have C. diff there, it gives it a better chance to kind of outcompete and proliferate more than what we want it to. All right, so that's really the number one risk factor. Also, just being older, being in some kind of long-term care facility or having a hospital stay, a weakened immune system, that's kind of just a catch-all. And if you've had C. diff before, you're more likely to get it again. Okay, so those are some of the risk factors. The spores are spread in feces. So this is the one exception. Remember last time I said organisms that sporulate will do so in the environment and not in the body. Okay, this is the one exception to that. So C. diff will, again, sporulate in the host. So that's the one exception to that. And so if you are maybe cleaning up after a patient that had C. diff diarrhea, you've got to gown up and, put the face shield on and gloves and everything. That is to protect you, but more so, if you're not on antibiotics, again, there have been cases, but it's really rare for you to get C. diff if you haven't been on antibiotics. It's mostly so you don't carry it to the next patient's room that is on antibiotics. And, we don't want to spread it that way. So the spores, again, they're spread in feces, but again, a lot of us have it in us already. Okay, usually if you're healthy, you fight it off, even if you are exposed. Okay. So I mentioned that it causes diarrhea, a very persistent diarrhea that's difficult to get rid of. A lot of strains are, or at least some strains are antibiotic resistant, so they don't respond well to antibiotics, and it gets to be kind of a long-term chronic, month after month thing that some people just can't get rid of, and contributes definitely to their mortality. But it can be mild anywhere. A mild C. diff diarrhea is just an explosive watery diarrhea. In severe cases, it can actually cause what's called a pseudomembranous colitis. So that's what we see here. This is a picture from an endoscope, I believe, and you see these yellow spots. That's the pseudomembrane. That's from inflammation. So yeah, lots of inflammation there. That looks just terribly painful. And it can potentially perforate the intestine. And you can get kind of a bloody diarrhea as well. Okay, so you get a perforated intestine, right? And then all those microbes is, spilling out into the abdominal cavity, and that's a life-threatening situation. Okay. In terms of virulence factors, I don't have a lot to talk about here. There's a couple of exotoxins. Remember, it's a gram-positive, so there's no endotoxin. This enterotoxin, that's just a toxin that acts in the intestine and just causes a change in the water homeostasis. Cytotoxin, killing these cells in the of the large intestine. In addition to that, fimbriae. Remember, fimbriae are what's going to help the organism attach to those cells of the large intestine. Okay. In addition, so I guess, I don't know, I was going to mention this before when we talked about how it's transmitted, but it's supposed to have, I haven't experienced this myself, but it's supposed to have a really distinctive odor about it that's really, really bad. I've heard it called the whiff of the diff, that some nurses and doctors can identify whether a patient has C. diff diarrhea just by smelling it. So you know what you're signing up for, those of you that want to treat patients with this. So what do we do for treatment? Usually the first thing we try is an oral treatment of vancomycin. And usually vancomycin was one of the antibiotics that we typically would give by IV or injection. But in this case, in order to deliver it in high enough concentration to the large intestine, we give it in pill form. And we always try, if not one course, 2 courses of that. And most of the time that will get rid of this. For some patients, though, they might have an antibiotic resistant strain, and that doesn't do the job. So this other treatment that some of you may have heard of is going by the name FMT, and that stands for fecal microbiota treatment for cases where antibiotics don't work. And so that is what it sounds like. They used to call it fecal transplantation. They used to take a donor's feces and put it up into the patient with an endoscope. And the idea here is to repopulate the large intestine with that good variety of microbes that are just going to outcompete the C. diff. Okay, and this has a really high rates of having it work in the 90s. More recently, they've been able to do this without using an endoscope. So they've been able to put the donated feces in a capsule. When they first started doing it, the only capsules that would dissolve at the right time were clear. So unfortunately, patients had to swallow these, clear capsules with fecal matter in them. Like they have to take like 10 of them at a time for like 3 to 5 days. Like it didn't sound pretty, but it made them feel better. So in the end, it was worth it. You have to freeze them so that those live organisms are okay. Now they've got it in capsules that just look a little bit more appetizing. I'm sure they're still pretty large. Some of you that donate plasma, hey, this is another option to be a fecal donor. So you might want to look into that. There is a company in Massachusetts, I know, that you can sign up to do that, but to be screened really carefully. Incidentally, I've heard these called crapsules. Appropriate, but don't try this at home. The only problem with this procedure is that they did have at least one patient die after having this procedure because he ended up getting a strain of antibiotic resistant organism that's from the donor that wasn't causing any problem in the donor, but the person that took it was more fragile health, and it ended up killing him. So that kind of slowed down the number of FMT procedures that they were doing, and now we screen donors as carefully as we can to try to avoid that. I just looked up, let's see if I can get this to come up. Before I came in here, the whole problem is that, this is a procedure that it doesn't really cost very much. So they were having trouble figuring out, like, how do we bill this? Like, nobody's making any money on it. So the FDA hadn't approved it until just a couple of months ago. Oh, there it comes. All right. So this was, as of December, the FDA approved this FMT therapy and issued some guidelines. So that may mean that this becomes a little bit more common. And I see now that there's a pharmaceutical company that is actually, has got the brand, they've got this just regulated more. So somebody's making money off of fecal donors now. So it may become more and more common. There. All right, so when the antibiotics don't work, we try to repopulate. Okay. All right, so moving on. So I've tried to give you a kind of hint as to where you'll find that pathogen in OpenStax and in the homework. In this the mycobacterium are under bacterial pathogens of the respiratory tract. So you should find that in 21.2. It's a little harder to, again, the book has presented it a little bit differently than what I am here. But mycobacterium, of course, we'll finish off our gram-negative, gram-positive pathogens. Sometimes mycobacterium is considered gram-positive because it weakly stains gram-positive, but most of the time we consider this a acid-fast organism, which is kind of neither gram-positive nor gram-negative. It doesn't have an outer membrane, so it's definitely not gram-negative. So sometimes they just group it in with the gram-positives. But we'll talk about why it's acid fast. Remember, it has this kind of waxy cell membrane. There's a lot of different species known. Most of them are considered biohazards, but the most famous ones are Mycobacterium leprosy, which causes the disease known as leprosy or Hansen's disease. There's actually a couple different varieties of that. The Lepratomus leprosy is the variety that causes basically body parts to fall off. It's an organism that grows and reproduces better when where the body temperature is a little bit lower. So fingers, nose, ears, and then patients eventually die of infections and losing body parts. Very treatable now. It's, you know, we have drugs that will treat it now, but you have to catch it kind of a little bit earlier on than that picture there. Mycobacterium bovis, that's tuberculosis in bovine tuberculosis. So not just cows, but hoofed animals like elk and deer and moose. When humans can get bovine TB from drinking unpasteurized milk, but it's a little bit milder form of TB. And then the one that we'll spend all our time on here is Mycobacterium tuberculosis. That's the organism that causes human tuberculosis, sometimes referred to as consumption or the White Death. It's been called the most successful pathogen of mankind. Okay, so back in 1876, when Robert Koch identified the infectious agent for tuberculosis, it killed about 1 out of every 7 deaths were due to TB. So this is something that mankind has dealt with for thousands of years. And it's still out there. We thought that we might eradicate it when we came out with the first antibiotic to treat it, and that hasn't happened and is not likely to happen anytime soon. It's estimated that 1 in 4 people worldwide are infected with TB. Now we've revised that. In 1999, we were saying 1 in 3. Now it would have been a little better, at least we say 1 in 4. We'll go through the progression of the disease. That doesn't mean that 1 in 4 people are infectious. It can kind of go into this stalemate form. So most of those people, again, have kind of a latent TB where it's not infectious, but still it's out there. Not so much in the US. You can see from this map, like the blue is where the incidence is really low. It's mostly sub-Saharan Africa and areas of Southeast Asia where the incidence is quite high. And as of 2021, about 1.6 million died of TB. So again, we don't hear a lot about it in the US. Here's some figures just from the US. The number that is usually thrown around is we have about 10,000 cases a year. A little bit less of that in 2020, I guess. You can see we, of course, this has been coming down since the 1940s when we came out with the antibiotic, but we had kind of a peak there, which corresponded roughly with the AIDS epidemic. And now that we're getting better at treating HIV incidence, controlling HIV a little bit better, incidence is getting lower. But you know, if we're going to try to eliminate this disease, it's something that we only pass around for human to human. We would have to have way lower cases than that. So we're not there yet. Okay, so who gets it? How is it transmitted? Like I said, this is humans only. Okay, so it's not zoonotic. Again, we can get the bovine form, but that's usually much more mild. And the transmission is considered airborne. And so we would say droplet nuclei. Okay, so they say not just sneezing, or even if you kiss somebody, that's not likely to get it that way. It's from the teeny tiny droplets that are suspended in the air for long periods of time that we breathe in. Okay, so if we swallowed the organism, it wouldn't do us any harm. We have to breathe it in. It has to get kind of into the depths of our lungs for it to infect us. So anybody can get tuberculosis if you're, if you're healthy. There are some risk factors. Obviously, if you have HIV and it's not, replication of that is not being controlled well. Diabetics are a lot more prone to get lots of different infections. Couple reasons: they have poor blood flow to their extremities, so their innate cells are not getting where they need to go to fight off infections. And if I should say, diabetics that are not controlling their blood sugar well, if you have more glucose in your tissues and blood than what you should, then that's just more food for pathogens to have access to. So okay, and just people living in congregate settings, so jails, for example, we see a higher incidence of TB in jails. And it really depends, this little cartoon down here, it depends, whether you get TB, how close have you been to the person that's coughing, that's ill with it, how close have you been, how long a time have you spent near them. So there's lots of other factors that kind of go into whether you're actually likely to get sick with TB or not. Virulence factors, we'll mention a few, primarily that acid fast cell wall. It's a complicated structure. We mostly just talked about the main feature, that mycolic acid, which, remember, is a long chain fatty acid. So that gives that cell wall kind of a waxy property. So when you think waxy, you think kind of impermeable to a lot of things. Okay, so a lot of antibiotics can't penetrate through that outer cell wall. We have a very short list of antibiotics that work here. It does protect the organism out in the environment. If a person coughed out a Mycobacterium tuberculosis bacilli, it would probably stay viable for weeks on some surface. Compare that to like maybe a day for an enveloped virus, or two. So again, keeps it from drying out. When it's phagocytized, it often will resist digestion, or sometimes the phagolysosome doesn't fuse with the phagosome even in the first place. Let's see, what was the other thing? And it's especially, in fact, this organism actually grows in macrophages. Okay, so the very immune cells that should be fighting it, that's where the Mycobacterium actually lives and reproduces preferentially. Okay, so it keeps from being digested and then reproduces in those macrophages. The cell wall does make the organism grow really slowly. So how slow does it grow? If you compare the doubling time of a Mycobacterium tuberculosis to E. coli, E. coli doubles about every 20 minutes. The generation time is about 20 minutes. For Mycobacterium tuberculosis, that time can be around 24 hours. So to grow up colonies on a plate, it will take weeks and weeks to grow up some Mycobacterium tuberculosis rather than just overnight. So slow to grow, also slow to die, as we'll see when we think about killing it. Taking drugs, it takes a long time to kill it. Other virulence factors, we know that, before I move on to that, I wanted to just kind of review when we do an acid-fast stain, especially for those of you in lab, we use a dye called carbofuchsin that has phenol in it that can penetrate through that waxy cell wall. So that's the kind of hot pink color there. And then that holds on to the dye even when we wash it in acid dissolved in alcohol. Okay, sorry, acid dissolved in acetone. Okay, so that's what makes it acid fast. It holds fast to that dye even when it's washed in acid. We add a blue counter stain for anything that's not acid fast. That's why you see all those blue smudgies in that picture. Okay. Other virulence factors, there's a glycolipid that is referred to as cord factor. And for a long time, we didn't really understand what cord factor did, but when we made mutants of this organism without cord factor, they didn't cause any infection at all. And the cells didn't kind of clump together. So they're really small bacilli, and they tend to kind of cord together like this in kind of a characteristic way. You know, what significance does that have? Well, we think it probably has some role to play in inhibiting that lysosome to fuse with the phagosome, helps it survive inside phagocytic cells like macrophages, and probably has some role in inhibiting the migration of other innate cells to the site of infection. And there's other mechanisms that when you try to look up virulence factors, it gets really complicated really quick, and there's a lot that we still don't understand with this organism, but mechanisms that help it survive inside that macrophage, like keeps the phagosome from acidifying. And I mentioned briefly that macrophages kind of operate on two levels, kind of a normal level where they go around just kind of cleaning up dead cells in the body, and then an activated level where they're you know, working even harder, and it keeps macrophages from being activated into kind of that next level. And then one more thing that ties into something we talked about a while ago, we think that it's really important that Mycobacterium tuberculosis are able to, a way that they're able to survive the nutrient deprivation that they have to endure when they're inside a macrophage, is to use the glyoxylate shunts. Okay, so we talked about this a long time ago. Remember, this is a variation of the Krebs cycle that not only some bacteria use, but it's prevalent in fungi, it's prevalent in plants, but humans are not able to use this pathway. So if you remember back, you know, a couple of months ago, we were talking about metabolism. This was the glyoxylate cycle, or the glyoxylate shunt figure that I showed you. If you remember the main idea there, they found that when Mycobacterium tuberculosis is inside a macrophage, they upregulate the production of this enzyme here, isocitrate lyase. So that's how they know that they're using the glyoxylate cycle. And with this enzyme, they peel off this 2-carbon glyoxylate, and then they can make more malates in order to make sugars. Whoops, sugars. Okay, so this is an anabolic pathway, meaning it's building molecules up, right, to synthesize, we'll say carbohydrates. Okay, so instead of losing a couple of carbon dioxides and losing all of their, so they don't lose those carbon dioxides when they take this shunt off. Okay, so they can conserve that carbon and make it into other sugars. Okay. So again, they think that pathway is really important in the pathogenesis of Mycobacterium tuberculosis. Okay, so let's follow the disease progression here. It's kind of a complicated progression. So if we start with the bacilli being inhaled. So tuberculosis can infect a lot of different tissues in the body, but it always starts in the lungs. Okay, so we inhale it and it infects the alveolar macrophages. We have a lot of macrophages controlling the very distal ends of those bronchial tubes. And they replicate freely in the macrophages there, and patients are going to experience kind of mild flu-like symptoms. Okay, and at that point, some people, I don't know, a good percentage of people actually fight off the infection. It's a strange thing. They say that this organism is not particularly infectious, so it's hard to understand how it's been so successful, going back thousands of years. But a lot of people actually fight it off at that point. But for people that don't, they are usually able to at least form a little bit of a stalemate with the bacteria. And that's because these granulomas that we call tubercules form in the lungs. Okay, so it's just kind of a specialized granuloma that we have a special name for because it's characteristic of tuberculosis. So tubercules. What's a tubercle? It's basically in the center, you have a lot of live cells, but then deposited outside of that are lots of dead cells. There's the structural protein collagen deposited around that. So it's kind of just a tough, abnormal kind of aggregate there in the lungs. And again, this is kind of a stalemate. So our immune system is not winning and the bacteria is not winning. We kind of form a truce. Okay, contained but not conquered. All right, and there are other ways that we sometimes describe those tubercules. Sometimes we call them GON complexes. That's when they get a little bit more calcified. And they show up a little bit better on X-ray. Sometimes we call them GON complexes. Sometimes we describe them as having caseous necrosis. A med student once told me there's actually 7 different kinds of necrosis, death of cells. So this is one that's pretty, I don't know if I think it's unique to tuberculosis, but caseous necrosis, that means kind of a inside of the tubercle, it has kind of a cheese-like consistency inside the tubercle. So it gets kind of runny and kind of yellow looking in there. So there's different ways that we describe those tubercules. But at this point, when there's a tubercle formed, the patient is not infectious. Okay, so again, it's kind of a stalemate. So, most of the people worldwide that are infected with TB, they have these tubercules in their lungs. At some point, though, that live bacteria in the center of the tubercle can actually rupture out of whatever the calcified part around it. Okay, now at this point, when that happens, whoops, uh-oh, what did I do? Yeah, okay, I must have bumped something. Let's try this again. Start the video. Oh, I'm still recording. Okay. Sure. No, I don't want to. Okay, come on, share my screen. Don't show me. Okay. Let's try this again. Just thinking how few problems I've had lately. Technology. Yay, we're back. Okay. Good. So when those tubercles rupture, okay, now the patient is infectious. Okay, and interestingly enough, only about 10% of the people that have these tubercles will it ever activate again. So that just makes it, complicated to figure out, well, do you treat somebody that has tubercles or you don't? You have to look at the patient's history and the likelihood that, maybe this will happen. When does it reactivate? Sometimes it's when the patient becomes immunocompromised, maybe they're having chemotherapy, or maybe they just have another infection, or maybe they're just aging and their immunity wanes. But sometimes we don't really know. So, but only about 10% of the time do those tubercles rupture. And now they're starting to get some symptoms like a productive cough, not just productive in terms of mucus, but you can have a bloody sputum. They're having chest pain. And then those symptoms will progress into what we kind of associate, when we use the term consumption. The patient is being consumed. They're exhausted. They have a fever. They have trouble sleeping. They're pale. That's where the term white death comes from. They have this ghostly like white pallor because their tissues are not getting enough oxygen. Okay. Still usually treatable at this point when it's reactivated. And we certainly do want to treat it now. The mortality is about 60% if it's not treated. Okay, so still people could even heal on their own. But when it disseminates, or sometimes we call this miliary TB, I think that's kind of a outdated term. Millet, that's named for millet seeds or kind of small lesions. But if it disseminates, that just means now it's not just in a patient's lungs, it's in the bloodstream and it's moving to lots of different body tissues. We can have loss of kidney function. It can infect the bones. It can infect the meninges, the covering of the spinal cord and the brain. So at this point, it's going to be 100% fatal without treatment, and even with treatment, a high degree of fatality. Okay, so patients can go into a coma, all kinds of problems. All right, so it's kind of a complicated disease. It's very complicated to diagnose it and treat it as well. So a lot of you may have had a tuberculin skin test recently, or if you're starting a new job or starting a new program. There's two different, like kind of the first thing that we do is kind of a screening to see if somebody has TB. And it can be one of these first two things. The most common is doing a tuberculin skin test. We just take a little bit of protein that's been taken from the organism and insert it with a needle underneath the skin. And then if we have ever been vaccinated for TB, have primary TB or active TB, then we'll have kind of a delayed hypersensitivity reaction to it. So you have to go back to the testing site, and they have to look at that lesion anywhere between 48 and 72 hours after you have the injection. And if you miss that, you have to have it again, and it has to be read in that certain time. And they're looking for some kind of lesion, like how big it has to be till they say that it's positive. It's kind of variable. But that's just the first step. That doesn't mean that you have active TB. It means that you've been exposed to it in some form. So it's only the first step. Becoming a little bit maybe more common is a TB blood test, but still I don't think they use it as a normal screening because it is more expensive. This is also referred to as an IGRA blood test. That stands for interferon gamma release assay. So we talked about alpha and beta interferon as protecting against viral infections. This is the type 2 interferon. So they do a blood test, and then they expose the blood to TB proteins. And if they find interferon gamma release, T cells will release interferon gamma in our blood when they are exposed to TB antigen. So if they find the interferon gamma, then we can say, oh yes, either you have primary TB or secondary TB. The advantage here is that prior vaccination does not cause a positive result. So it's a little bit better in that way, although it is expensive. So if either one of those first two steps are positive, typically next, they'll do just to kind of rule out TB, they'll do a chest x-ray. If a patient has the tubercles for primary TB, then they're usually a little bit lower in the thoracic cavity. For reactivated or secondary TB, they're usually a little bit higher. So they can kind of look at the position and know. And of course, if there's no tubercles, then you can rule out that the patient has primary or secondary TB. If they do find those tubercles, then they'll try to do some microscopy of sputum. So not just saliva, but you got to really cough something up get the patient to cough up something from their lungs. And they'll either do some acid fast staining, or probably more likely, they'll use some kind of fluorescent microscopy where they can tag very precisely that mycolic acid, and then only cells with mycolic acid will fluoresce. Okay, so it's a really a little bit easier actually to see if there's live bacilli. And if those bacilli show up, then you know that the patient has active TB. So again, it's complicated to figure that out, and kind of what complicates that is in areas of the world where people routinely get vaccinated. Treatments. So we came out with streptomycin. That was Selman-Waksman. I think 1949 is when that was approved, and that was a miracle drug. That was the first drug that we had that worked against TB. We talked about some of the side effects of that drug. Remember, hearing loss was one of the side effects. So I had to dose it pretty carefully. 1946, I said there. We don't use that anymore because most strains of TB are resistant to it. So today, there's two drugs that we always use, isoniazid and rifampicin. Remember, isoniazid was the one that targets mycolic acid synthesis. So that's a pretty narrow spectrum for any mycobacterium. Rifampicin was the one that made your urine turn orange. It was an orange pill. It targets RNA polymerase. And then we always use a couple of others. I didn't add them here because we hadn't talked about them before. One of the drugs, we're not even quite sure how it works, but it does kind of keep the TB from replicating. And that's just for like normal garden variety TB. Patients have to be on these 4 drugs for at least 6 months. And that's taking quite a lot of pills every day. That's why I showed this picture down here. And side effects are common. So things like nausea, tiredness, rash, headaches. Okay, so that's why they will often have a healthcare professional come to a patient's house and watch them and make sure that they're taking their TB drugs. Because maybe after a month or two, they're going to start feeling better, but they from the TB, but then they still have this, effects of nausea and things from taking the drug, and they want to stop taking the drug. But we want them to take a full dose to get rid of all those bacilli, because we have a terrible problem with resistance. About 10% of the new TB cases that are diagnosed are multiple drug resistant. So these first line drugs don't work anymore, and they will find other drugs. Sometimes they'll try streptomycin again to see if that works. And usually they have to be on these drugs for 2 years. And it's, 20 plus pills a day. Again, side effects associated with that. And then anywhere from 1 to 3% of new cases are extensively drug resistant, meaning the second line drugs aren't working either. The good news is we do have a drug that was just approved in 2012 called bedaquiline, which has, it's the first kind of new mechanism to treat tuberculosis that we've come out with in like 50 years. And it targets the Mycobacterium ATP synthase. Okay, but still we give that in combination with some other drugs, and again, throw a lot of stuff at it, and hopefully something works. Okay, so, and there's, again, just a huge population, with the HIV epidemic, we had lots more people getting TB because their immunity was low, and again, just lots of trouble with resistance. So do we have a vaccine? Yes, we have a vaccine. It seems like let's try to prevent it to begin with. The vaccine is called the BCG vaccine, which stands for the two Frenchmen that first made it, Calmette and Guerin, and they called it Bacillus, or in French, Bacille Calmette Guerin. They came out with this vaccine in 1921, and it has been changed very little since then. So the World Health Organization just came out, last year and said, this vaccine is 100 years old. We can do better. We need more research into this to make a better vaccine. Part of the problem is we don't have a good animal model for tuberculosis, for human tuberculosis. So that's part of the problem. But this is an attenuated vaccine. Basically, they took a strain of Mycobacterium bovis and just passaged it over and over and over and over and over again, and it kind of lost some of its virulence factors. It works pretty well in children. Unfortunately, it doesn't work as well in adults. And the protection lasts somewhere between 5 and 15 years, and then people have to get booster doses. So they're really hoping to develop a better vaccine. We don't routinely vaccinate in this country just because it's not all that common. And then if you do end up getting it, it just complicates the whole diagnosis process. But people that are vaccinated tend to have a little bit milder form of TB if they do get it. Okay. All right, questions, anything about TB? All right, let's move on to some gram negatives. So I'm going to kind of skip over gram-negative bacilli, because we've worked with a lot of those in lab, like E. coli is a gram-negative bacilli. What we haven't worked with in lab are any gram-negative cocci. So when you gram stain in lab, all the round cells are purple and all the rods mostly are pink, but there are gram-negative cocci. We don't work with them in lab because the Neisseria, at least, are very difficult to grow in the lab. Let's see. They're actually, I'll come back to that. They're actually, we consider this genera Neisseria, we consider them fastidious. That means they're just difficult to grow in the lab. They have specific growth requirements that they need a special media to grow on. So if we streak them out on tryptic soy agar or nutrient agar, they wouldn't grow. And in addition, when they run out of glucose and kind of enter their stationary phase of growth, they tend to autolyse. They tend to die. So they're hard to keep alive. But Neisseria is the genera of gram-negative cocci that are sort of clinically interesting. In other words, there's two species that typically cause disease. They usually present as diplococci. Okay, so if you can kind of see, they say that this picture is a gram stain. I don't know whether they're using like really concentrated safranin or something, but that's not how that red color, we don't usually see that when we gram stain. But you can kind of see, if you look really close here, that these are typically pairs of cocci growing there. And that's very typical. They're not motile, and they're aerobes. Okay. So the two different ones that we're going to talk about, the Neisseria gonorrhea, by far the hardest organism to spell, as if Neisseria wasn't hard enough. So this is the causative agent of gonorrhea. And we also sometimes refer to this as the gonococcus, because whoops, let's spell that right. Okay, that was what the genus used to be called before we changed it to Neisseria. So the gonococcus. And Neisseria meningitidis is the causative agent of meningococcal meningitis. And that used to be called meningococcus. Okay, so again, those are outdated, but sometimes you kind of read that in the literature. Okay, so we have talked about, we're going to talk about gonorrhea first, but we've talked about several different organisms that cause meningitis. So the Neisserial meningitis, again, we call them meningococcal meningitis. There's also viral meningitis, and usually viral infections they're harder to treat, so they're a little bit more serious, but that's not the case because the bacterial meningitis produce different kinds of toxins that make it much harder for us to deal with. Viral meningitis usually isn't terribly serious. It's all the bacterial ones that we need to worry about. So that's kind of unusual. All right, so you'll find this pathogen in the section in your book that talks about bacterial infections of the urogenital tract. Okay, and this is a true pathogen. It has a very low infectious dose. It doesn't matter how healthy you are, you can get it. The transmission of it, this is a sexually transmitted infection. Okay. So it's going to be carried by the sperm and vaginal fluid. And just going to target different cells based on anatomy. So for males, the urethra is targeted. And females, the cervix. Okay, so, and interestingly, a male with gonorrhea is much more likely to give it to their uninfected partner. About 50% of uninfected females would get it from an infected male. The transmission is a little bit less efficient the other way. So a female with gonorrhea would only give it to her uninfected partner about 20% of the time. Okay, so the bacteria kind of hitches a ride on the sperm. We also have mother to child transmission. Okay, during birth, during a vaginal birth. And most commonly that would affect babies by this gonococcal eye infection, ophthalmia neonatorum, which causes these kind of crusty lesions on the eyes and can lead to blindness. So for that reason, almost as soon as babies are born, they get eye drops put on them. And we used to use silver nitrate. Now we use antibiotics. I think it's erythromycin. It's a little bit less irritating than the silver nitrate was. You know, they could only treat babies whose mothers have gonorrhea, but sometimes mothers wouldn't admit that they have gonorrhea, or sometimes they don't know. So for that reason, they just treat all newborn babies to avoid this eye infection that can, again, lead to blindness. All right, so what virulence factors make this organism successful? The big one is fimbriae, and those fimbriae have some kind of unique properties that make it able to persist in the host. Usually we call it fimbriae. Remember, these two terms, pili and fimbriae, are sometimes used kind of interchangeably. They're both made of the same protein, pilin. Oftentimes, for whatever reason, when we're talking about gonorrhea, we call them pili. I think your book calls us some fimbriae, but the genes are like pill this, pill that. So I don't know, whichever you call them, pili or fimbriae, they're for attachment. So the cocci can attach to the membranes of the genital tract. Okay, and there's two very interesting properties these, fimbriae or pili have that make them successful in evading our immune system. They can do something called antigenic variation. And that just means that the composition of the fimbral proteins changes. Okay, so we're not going to get into the mechanism of actually how that happens. If you're more interested in that, keep taking microbiology classes. They talk about that in the upper level medical bacterial pathology class. But it's a kind of a unique mechanism that not a lot of organisms use to kind of change out their amino acids in these proteins. So the other organism that is really good at that is the Lyme disease, Borrelia burgdorferi. But gonorrhea, Neisseria gonorrhea does this really fast, so fast that when we mount an immune response to those fimbriae, so it's on the outside of the cell, they're proteins, they're really antigenic. By the time, it takes maybe 10 or 14 days to make antibodies. Once we do, those proteins have changed and the antibodies aren't any good anymore. So then it takes a whole nother, 10 or 14 days to get, the right B cells and T cells interacting. And then as soon as it does, now those aren't good anymore. So it really makes that organism good at persisting. And it can do something else that we call phase variation. And that just means it has the ability to turn it on and off the expression of the pili. So the pili, or what we call them fimbriae or pili, they detach. Okay. So basically the immune system follows those pili, the antibodies follow the pili, and meanwhile the cells are going off and reproducing. So it's kind of like, if you're getting chased by a polar bear, what do? You throw down your mitten so the polar bear stops and smells the mitten and you get away. It's like the exact same thing. Right? Okay. So antigenic phase variation, basically it's due to those two things that we don't have a vaccine to gonorrhea. It changes too quickly. Patients can get gonorrhea over and over and over again. The memory that we have is not to the right antigen because it keeps changing all the time. So some other things like some exoenzymes, it makes a protease that cleaves IgA. Remember, that's the most prevalent antibody on the mucous membranes. Okay, so, and then also LOS, that is a variation of LPS, lipopolysaccharide. Lipooligosaccharide basically just has a little shorter sugar chain there, but it's still got that lipid A endotoxin. Okay, that lipid really gives that outer membrane a really kind of impermeable aspect to it. So, you know, we talk about how it's causing inflammation, and you would think, oh, we wouldn't want to cause inflammation because that helps us fight the pathogen. But again, it has the role of making that outer membrane pretty impermeable to a lot of things. So remember that endotoxin causes things like kind of low-grade fever, inflammation, and also some, we'll say, inappropriate blood clotting. And we see that manifests a little bit more in the Neisseria meningitidis, but it will cause clotting in places that doesn't need to be clotted, and that kind of uses up all the clotting factor in those places, and other places in the body that need to clot the blood, then it's not able to. So that's, again, endotoxin. That's part of that little kind of variation of LPS. Okay. No exotoxins per se for this organism. All right, so what are the symptoms of gonorrhea? They're quite different depending on whether you're male or female. Males have described it as it feels like you're peeing fire. And your book kind of downplays that and says that, well, a lot of males don't have symptoms, but that's not what I read from lots of other sources. So my understanding is that most of the time males do have symptoms. Not only does it hurt when they urinate, but they have kind of a pussy discharge, and it's very painful. So that means they go and they seek treatment. And right now, gonorrhea is like 99% treatable. We have worries about resistance growing, but for now, it's very treatable. If it's not treated, it can lead to infertility due to inflammation in the prostate or epididymis. And it also has the potential to disseminate like in the bloodstream and cause all sorts of crazy things like arthritis, rashes, endocarditis, which is infection in the heart valves. Okay, so and it sounds like from some articles that I read recently is that disseminated gonorrhea is kind of the incidence, and that is kind of ticking up. Females, at least maybe half the time, they don't have any symptoms at all. And that's not necessarily a good thing, because that doesn't mean that the organism is not causing damage. When females do have symptoms, it often can mirror something else, like it seems like it's a urinary tract infection, or it seems like it's a yeast infection. So, you know, they would go, you hopefully would go to the doctor for a UTI, but if you think you have a vaginal infection, you'd probably try to treat that yourself, and then it wouldn't do any good. So in females, when they have an infection and they don't have symptoms or they don't get treatment, then that can lead to some more serious complications like pelvic inflammatory disease. which is inflammation of the cervix and the uterus. And that causes some kind of painful cramping and fever. And gonorrhea is not the only thing that can cause that. Chlamydia can cause that as well. There's probably other things. And if that's not treated, that can lead to infertility. So pelvic inflammatory disease can lead to ectopic pregnancies, where the egg implants outside of the uterus, and that can potentially cause infertility. Scarring of the fallopian tubes. Okay, so we want to treat this if we know about it. Incidence is ticking up. It's not as high as it was during the AIDS crisis. When everybody was worried about AIDS, then there was, more, abstinence or safe sex going on. People are less worried about AIDS now, I think. So the cases have, as of 2019, cases have doubled. This 600,000 figure is probably quite a bit of an underestimation, again, because people don't know necessarily that they have it. Undercounting is pretty likely. Most of you are in the most common age group. The more sexual partners one has, the more likely they are to contract this. It is the second most common and sexually transmitted disease behind chlamydia. But oftentimes people will have chlamydia and gonorrhea at the same time. Okay. So let's see, what else? Oh, worldwide, 82 million cases of gonorrhea in 2020. Okay, and when we think about treatments, there's certain areas of the world, especially Southeast Asia, where we seem to have some of the drug-resistant varieties coming from. Like I said, right now it's still mostly treatable. Usually we use ceftriaxone, which is a cephalosporin. Has to be injected. It's one of the later generation cephalosporins. But if you look back kind of over the years, this is a kind of an interesting 2-minute video that the CDC put out. We used to treat gonorrhea with a shot of penicillin, and that did the trick. But then it became resistant to that, and then we switched to this drug, and then it became resistant to that. So just kind of all the different, things that we've had to change. So this resistant to it is a growing concern. There's some strains, especially haven't shown up very often in this country, but kind of superbugs that are resistant to most everything that we have. And a lot of strains are resistant to at least one of those things. So that's a problem. One article that I just read this morning said that they think one thing that might be contributing to that is when homosexuals have, or when there's, sex men between men and, involves the rectum or the oral cavity, that can interact with different microbiome, different organisms, and they share plasmids and resistance plasmids by horizontal gene transfer, and that might be something that's contributing to this, but it's just one of those things. Okay, do we have a vaccine? No, no vaccine. It changes too quickly. Again, diseases that we can get over and over again, we usually don't have good vaccines for. It's those things that you get once and then you typically never get it again, have good memory that we don't have vaccine. Okay, so I just want to start the Neisseria meningitidis because in a lot of ways this is similar, but of course we have very different disease pathology, for two organisms that are quite closely related. This is in the section that's gram-negative bacterial infections of the neural, what's the word I'm looking for? Brain and spinal cord, whatever. Okay, like Neisseria gonorrhea, I didn't mention that, but that is just a disease between humans. Okay, so the reservoir is humans. We think about 10 to 20% of us are carriers, which is kind of astonishing because it's such a severe infection for reasons that we don't understand. So this would be carriers kind of upper respiratory tract or throat. For reasons we don't understand, sometimes it gets into the bloodstream and then, causes inflammation, particularly in the meninges, which is this 3-layered membrane that protects our brain and spinal cord. Okay, transmission, droplet spread. So this is close contact. This is not airborne. So sneezing, coughing, sometimes by some fomites, inanimate objects like a drinking glass. Okay, so we see, epidemics at universities every once in a while, and often will focus around some party where everybody's sharing glasses. Okay, I think that we're about out of time, so we'll start with disease symptoms next time. Have a good weekend. And I think we're in good shape to cover what I was hoping that we would cover. So have a good day.